Okay guys, welcome back. We are starting CV diagnostics. This will be a lecture that I'll most likely do in two parts, part one and part two, just because of the length of the slides. So basically this PowerPoint is actually from Egan's chapter 10 is kind of how it's following, but I've also have used material from chapter 5 out of Desjardins, your cardiopulmonary anatomy and physiology book, the blue one. So I kind of like both of them. So Desjardins, I think in some ways is a little bit easier to read, though chapter 5 does cover more than what we go into because it talks about blood and plasma and stuff. So we'll go ahead and just kind of get started here. The first couple slides are our learning objectives, and then we move on to anatomy of the heart, where, for most of, actually for the first couple of lectures, I would say a lot of this should be reviewed, since now I have taught cardiac A&P, your first semester class. I didn't teach it to you guys. I think Melanie still had it, but I did just teach it for the first time to the A group. So as I went through that, I was like, oh, we hit a lot of this in, you guys have already hit some of this. So hopefully it should be reviewed. So that's kind of some basic stuff that we're going to go over here. All right, so just kind of talking about the basic anatomy, you should know by now it's a hollow, four-chambered muscle, lies behind the sternum. 2/3 of it lies to the left between the second, is your fill in the blank there, through the 6th rib. So the second through the 6th ribs behind the sternum. And the heart apex is your next fill in the blank, is at the 5th intercostal space. Okay, and remember that your PMI is your not Pima Medical Institute, PMI is point of maximum intensity, okay, and that's the strongest palpation of the heartbeat is found in the apex, okay. So, and remember, your heart, when you look at your heart or you think about it, it's kind of upside down then, so your apex is actually, pointed down. In fact, I do recall, I think, when I was teaching cardiac A&P, it points at your hip. Let's see, your left, I think it points in towards your right hip. But anyway, it's upside down. Okay. All right, this is kind of part of it where it gets just a little bit confusing. We're just going to kind of take it slow. I find it helpful to draw out the layers myself. In fact, when I teach it, I do put it on the board, and I believe the next slide here is coming up that has a picture, because I have on my notes, see PowerPoint for pick. But just to let you know, if you go on page 220 on Desjardins, you'll find a nice cross-section picture. So I kind of like to, and I'll tell you kind of how I labeled mine, but I basically followed the picture. But I think some of it gets confusing because there's terminology that's used for, like for example, for the visceral layer is also known as epicardium. So just get these layers down because they are definitely on your test. Okay. All right, so overall you look at your heart and it's enclosed in a sac, okay, and that is the pericardium. Okay, so that's a doubled wall sac that actually encloses your heart. Okay. And then the pericardium structure is made out of this outer layer, okay, fibrous being your fill in the blank there. Fibrous pericardium is kind of like that sac. It's your outer layer. It's not non-elastic. Thought I had fixed that. Not inelastic, non-elastic. That surrounds your heart. And then serous pericardium. Okay, so that's beneath then that outer layer, so you're going deeper. Okay, so you have the parietal layer, okay, which is the outer then of those two layers. Okay, so you have the serous pericardium, which consists of two layers. The outermost of those two layers is the parietal layer, which is then basically the inside lining of that fibrous layer. So it's right underneath that fibrous layer. And then the visceral or epicardium. Okay, so be familiar with both of those terms, and that is the innermost layer of those two layers, and that is what covers the heart and the great vessels. So I think, yeah. So this picture here, this is a little bit different picture. I forget where I got this one. I think I just got it online, or maybe I got it from. Actually, I think I got it from Egan. But yeah, so that's a cross section, but there's another cross section picture, and I kind of like that one better on page 220 in Desjardins. Okay, so it shows the fibrous layer again being like that real tough kind of, doesn't stretch, thicker layer, and then you have the parietal layer of the serous, and there's the parietal layer of the serous pericardium, and then you have that pericardial cavity. Okay, so there's fluid in there, and then you have the other two layers of your serous pericardium, which then consists of the parietal layer and then the epicardium. Okay, and then the heart muscle itself then also has layers, and we'll talk about that here in the coming slides. So try to get those layers down. If it helps, I think it helps just to kind of draw them out yourself. All right, so the pericardial fluid then is that thin layer of fluid that actually separates the parietal and the visceral pericardium. Okay, so that's what helps it pump easy. You don't have as much friction. But occasionally you can get fluid, or you can get inflammation in there, so you could have a pericardial effusion, which would be the fluid buildup, or you can get like a pericardial rub, where you actually can kind of hear a different heart sound. That's going to actually be like some inflamed area, so it doesn't slide as easily because now it is thickened, and it creates that rub, it creates friction. So that's like a sign of some type of infection or inflammation of some sort. So when we have that buildup of the fluid, then we can have it can develop into a tamponade. If you have enough fluid, okay, it would have to be a fair amount of fluid to actually affect the pumping capability of your heart. Okay, so that's what a cardiac tamponade is. And that's not the only cause of a tamponade. You can also have a tamponade from like a trauma, from bleeding, from post-op heart surgery. If you've got a bleeder or something in there and you start dropping blood pressure, you have to look to see if there's a leak. And then the pericarditis would then be that inflammation of the pericardium, where you would hear maybe that rub that I was referring to. All right, so now we're talking about the heart wall. The muscle layer itself, or the actual, one of the layers is a muscle layer, but what is the three layers of the heart wall itself, not just covering the heart? I hope I'm not too confusing. Sometimes I should just stick to the facts. So the outer is the epicardium, okay, epi meaning outer, okay, epicardium, EPI, okay, and then your myo. So when you think of the prefix MYO, you should think of muscle, okay, so your myocardium, and then your endocardium, ENDO, endo meaning inner, okay. And then we have our four chambers. We have our atria and our ventricles. All right, just a couple different pictures here. Yeah, so I guess the apex that would be of the heart would be pointing towards your left hip. Yeah, that makes sense. I was like thinking about the ankles. It's your right hip or your left hip? Yeah, it'd be your left hip. So this is again out of Egan's. And again, chapter 10 out of Egan's. So just being familiar anatomically where the heart is placed. And this is again a cross section of the heart itself, kind of showing the chambers, the different valves, and we'll talk more about that coming up. Okay, so we're going to talk about the AV valves, which are the atrial atrioventricular valves. I don't know, we just say AV valves, and sometimes I don't think about the proper pronunciation. So we have the AV valves. So basically that's between the valves that are lying between the atria and the ventricles. Okay, so those are your tricuspid and your mitral valves. So your tri valve is on the right side. Okay, try before you buy. I'm sure Melanie probably told you that one. So tri, so if you think about the blood flow going to the right side, then the left side of the heart. Okay, so tri is on the right, and then bi, or your mitral valve, is on your left. Okay, so these help basically prevent a backflow of the blood into the atria. Okay, so I'll back up here a second. The first fill in the blank then is right atrium and mitral valve at the left atrium. Okay, and then your third fill in the blank then is atria. Okay, so the ventricular contraction forces these valves closed, which then prevent that backflow of the blood into the atria. Then the lower ends of the valves, and you might remember when you did your had the actual heart from the lamb hearts or sheep hearts or whatever we got for your cardiac EMP, you actually were able to dissect the heart and look at it. Remember the word chordae tendineae. Okay, so the lower ends of the valves anchor to the ventricular papillary muscles by that chordae tendineae. Okay, so that's a good test question. That's your fill in the blank there. So the papillary contraction during systole pulls on these chordae tendineae, preventing the valve reversing into the atria. So basically, I look at it as, oh darn it, I thought I put the picture in there. If you look at Google images of chordae tendineae, it reminds me of like a trampoline type of thing, where when you pull on those, or maybe a parachute's a better explanation actually. When you pull on the cords, you know, you tighten on them, then the valve itself kind of goes back up, kind of puffs back up, and that's what helps prevent the backflow. So you think of like a parachute, and when it, when you pull on those strings, it kind of puffs back up to prevent the backflow. Sorry, I thought I had a picture on there. I guess I'm going to have to correct that. All right. So this is another cross-section picture of from your textbook. Okay, so, oh, there's my picture. I knew I had it someplace. I'll just have to change the order of it. Darn it. Okay, so that's what I was trying to talk about. So how you can see on the right side there, when the muscles are contracted and they pull those chordae tendineae more taut, or the strings kind of more taut, then the valve kind of floats up, kind of pulls on it, and it closes it, and that's what helps prevent the backflow. I knew I had it someplace. Okay, cool, very happy. And then on the other side, you can see on the left side when it's relaxed, those tendonae are more slack, and then it allows the valve to be open then and the blood flow to happen. Perfect. All right. So we have also semilunar valves, and these are 3 half-moon shaped valves or cusps. They help separate the ventricles from their arterial outflow tracts, the pulmonary artery and the aorta. So they are situated at the ventricle exits to the outflow tracts, the arterial trunks. So you have basically your pulmonary valve, okay, that lies then between your right ventricle and your pulmonary artery, and then your aortic valve, which is between your left ventricle and your aorta. So pulmonary valve being your first one and aortic valve being your second one. Okay, so make sure that you have those, so kind of just review the blood flow of the heart might help you too, to kind of draw this all out again. And then what's happening during that blood flow, during when the heart's contracting. Okay, so during contraction then, the valves, the semilunar valves, are going to open. Okay, they allow that blood flow then into the arteries, out to the pulmonary artery and the aorta. And then during the relaxation part, okay, during diastole, the valves then close. Okay, that's preventing the backflow of the blood back into the ventricles. Okay, so when we have regurgitation, what's happening there is the backflow of blood through an incompetent or a damaged valve. Okay, so the backflow of blood through an incompetent or damaged valve. Okay, then stenosis then is a pathologic narrowing or constriction of a valve outlet, which then causes increased pressure. I would hope that you would think that. Okay, good old physics there. Okay, so I will quickly go over the fill in the blanks again on this slide. So your first one is valves open, and then the second fill in the blank is valves close. Regurgitation is the backflow through an incompetent, incompetent or damaged valve. And then stenosis is a pathologic narrowing or constriction which causes increased pressure in the proximal chamber and vessels. Okay, now we're moving on to the actual coronary circulation, because the heart itself has blood flow to it, and when we have coronary artery disease, we have a heart attack, that's what's being affected, is the coronary circulation. Okay, so the heart actually requires more blood flow per gram of tissue weight than any other organ besides the kidney. Okay. Alright, so we have two main coronary arteries come off of the root of the aorta, and that is the left coronary artery and the right coronary artery. Okay, again, like I said, a lot of this should be review. Coronary artery pressure becomes higher than aortic pressure during systole. Okay, so the coronary artery pressure becomes higher than the aortic pressure during systole. Okay, this prevents flow of blood into the coronaries. So the diastole part is where that's the most important part. Okay, that's where the blood flow actually to the coronary arteries happens, during that diastole part. So that would be your third, or rather your fourth fill in the blank. Okay, so quick review: we have left and right coronary arteries. The third one there, the coronary artery pressure becomes higher than aortic pressure during systole, and then diastole, during that relaxation part, is when the coronary perfusion is happening. Okay. So that's why it's important to slow down the heart rate. So because a high heart rate, if you're too high of a heart rate, you're not going to allow, well, there's a couple different things. You're not going to allow the ventricles to fill, so your cardiac output's going to drop. And then you also have decreased coronary artery perfusion when there's a high heart rate. And that can actually lead to, again, decreased overall cardiac output, which due to the less ventricular filling time. Okay. So the healthy heart muscle actually requires approximately 1/20th of the blood supply of the body to function properly. So that's a lot of blood. All right, so your left coronary artery is positioned underneath your aortic semilunar valve, and then the left coronary artery branches off into your LAD and your circumflex. Okay, so the left coronary artery provides blood flow then to the left atrium, the left ventricle, and the majority of the interventricular septum, half of the interatrial septum, and part of the right atrium. Okay, so your left coronary artery, your LCA, okay, so you can see it branches to the LAD, which goes between the left and right ventricles, and then the circumflex, which then goes around the left side of the heart between the left atrium and the left ventricle. Okay, but overall, that whole, on the top of that left coronary artery provides quite a bit of blood flow, again, to the left atrium, to the left ventricle, majority of the interventricular septum, half of the interatrial septum, and part of the right atrium. Okay. On the next slide, you'll just kind of see a little visual of the left coronary artery, and this again you can find in your Egan's book. Hopefully you have it with you. I'm sure you do. It's probably in the trunk of your car, maybe. All right. So on the right coronary artery, that goes around on the right side of the heart between the right atrium and the right ventricle. So then many small branches as the right coronary artery moves around the right ventricle. Okay, the right coronary artery then ends in the posterior descending branch, which then travels between the right and left ventricle. So the right coronary artery then provides blood flow to most of the right ventricle and right atrium, including your sinus node. Sorry. So provides blood flow to most of the right ventricle and right atrium, including sinus node. All right, so what happens when we have impairment of that blood flow? Okay, if our coronary arteries are damaged or thickened, or there's a blood clot or whatever, it's not a good outcome for us. So we have an MI, okay, you have your 1 MI, your myocardial ischemia. Sorry, I didn't mean to be confusing there. Myocardial ischemia, okay. So that's basically angina, okay, so chest pain. So that's where you're going to have a partial obstruction of the coronary artery. Okay, so myocardial ischemia. And then your MI, your big one, your MI, infarction, okay, is your myocardial infarction, okay. So infarction basically means death of the heart tissue, or death of whatever tissue. It could be infarction in your brain too, or your lung for that matter. So you basically have a complete obstruction of the coronary artery, okay. So an MI, infarction, is worse than ischemia. All right, so then we also have the coronary veins, okay? They're going to collect then the venous blood that's passing through the myocardial capillary bed, and they run fairly parallel to the coronary arteries. So your first set of fill in the blanks there is your great cardiac vein follows your LAD, and the LAD is left anterior descending. And then your small cardiac vein follows closely along with your RCA, or your right coronary artery. Left posterior vein follows your circumflex, and then your middle vein follows your right posterior descending. Okay, so you've got great cardiac, small cardiac, left posterior, and middle vein. And then these all then come together to form the coronary sinus, which empties into where? The right atrium. Okay, so they all come together to form coronary sinus, which empties into the right atrium. So the Bayesian veins, the Bayesian, T-H-E, like the, then B-E-S-I-A-N, lesbian, not that, is not, there's not another S there, the Bayesian veins. These drain some of the coronary venous blood into all the heart chambers. Those draining into left atrium and right ventricle bypass the lungs, and these create a anatomic shunt. And the normal anatomic shunt is about 2 to 3% of your total cardiac output. All right. Properties of the heart muscle. So the heart's ability to pump depends on initiating, conducting electrical impulses, and the synchronous of myocardial contraction. So this is made possible by key properties that the myocardial tissue has. Okay, one of them is called excitability. That's the ability to respond to stimuli. So excitability is your first fill in the blank. Okay, so the stimuli, we just want to talk about that for a second, is either electrical, mechanical, or chemical. Okay, so the excitability is ability to respond to stimuli, which is either electrical, mechanical, or chemical. Those are all different types of stimulation. Okay, and we look at electrolyte imbalances, and certain drugs can actually increase myocardial excitability and produce abnormalities in the electrical condition, which can lead to arrhythmias. Okay, so I'll say that again. I know you can rewind it, but electrical imbalances in certain drugs can increase myocardial excitability and produce abnormalities in the electrical condition, which can actually lead to arrhythmias. The other property of the heart muscle is, or the heart tissue, well, myocardial tissue is heart, or muscle, sorry, is your inherent rhythmicity. Inherent, I-N-H-E-R-E-N-T, inherent, and rhythmicity, rhythm, R-H-Y-T-H-M-I, city, C-I-T-Y, or your automaticity. Okay, so that is basically the initiation of the spontaneous electrical impulse, which mainly comes from your SA and your AV node, because those are the primary pacemakers. However, keeping in mind, sometimes we have these other areas that can start a heartbeat, and that's what kind of contributes to arrhythmias. And then conductivity spreads the impulses quickly. So abnormal conductivity can affect timing of the chambers of contractions and decrease cardiac efficiency. So if we have kind of a abnormal spread of that impulse, it can affect our cardiac output. And then contractility is your last one. Contractility is the contraction in response to electrical impulse, which is your primary function of the myocardium. Okay. One of the unique features of the myocardial tissue is that it cannot go on tetany, which means it cannot involuntarily contract or cramp up. So make sure you have all four of the, well, really five, fill in the blanks there. Your first one being excitability, your second one inherent rhythmicity or automaticity, conductivity, and then contractility. Okay. Refractory period. That's where, again, where it cannot go into the tetany or the sustained. That's the time period where the myocardium cannot be stimulated. Okay, the refractory period. That's kind of your it's lasts about 250 milliseconds, about as long as systole, as long as the contraction. So the heart muscle is kind of protected in that sense. That's part of the properties of heart muscles, where it kind of makes it unique. So the microanatomy of the heart muscle, we have the cardiac cells are the short, fat, branched, and interconnected. Oh, I gotta back up here just a little bit. So your I just want to make sure you got the fill in the blank. So your time period, myocardium, okay, cannot be stimulated, lasts 250 milliseconds, nearly as long as systole. I was looking at my notes, I didn't look up at the slide. Alright, so we have the cardiac cells, is your first one there under the microanatomy. And then your individual cell fibers are enclosed in a membrane called the sarcolemma, S-A-R-C-O-L-E-M-M-A, sarcolemma. And then each fiber consists of many smaller units called myofibrils. Myofibrils, M-Y-O-F-I-B-R-I-L-S, which then contain this repeated structures about 2 microns, which are called sacromeres or sarcomeres, S-A-R-C-O-M-E-R-E-S. Okay, that's just a bunch of terminology there. Alright, so here is a visual. Again, this is out of Egan's. It's the major structural features of the cardiac muscle fibers. And you can find this in Chapter 10 in Egan. So we're going to stop this PowerPoint. This is ending Part 1, and then Part 2 is going to start with the whole vascular system. Okay.